These are the lecture notes for Chapter 13, Regulating Gene Expression. Let me go ahead and set this slideshow up. And I guess that'll be good enough. Okay, so one thing I wanted to let you know is that there is a section 13.1, on prokaryotic gene regulation. We will not cover this topic. We will skip this topic. This is already a long, kind of complicated chapter that elaborates on transcription and translation, which we learned in Chapter 12. So we're just going to stick with eukaryotic gene regulation and then the section on gene mutations. Okay, so in the introduction, there is some information on how one of the ways that scientists began to become interested in gene expression was learning about gene expression in bats. And as you know, bats are mammals. Researchers have discovered that the immune systems of bats have evolved to basically ignore certain viruses. The viruses can attack them, but typically what will happen is that the immune system will start out by causing inflammation. And inflammation is the redness, heat, swelling, and pain that results when we're sick. It causes us to have allergic reactions, and it causes us to have the symptoms of being sick, basically. So bats really just don't get sick from these viruses. They don't have that inflammatory response. And the reason is bats have a higher tolerance for some mammalian viruses and infections than other mammals because they have a lowered expression of inflammatory genes. And what inflammatory genes do is they regulate that inflammatory response that occurs when we have an infection. Since bats have a lowered expression of inflammatory genes, they can be exposed to certain viruses but not exhibit the inflammatory response. In other words, they don't get sick. They don't exhibit the symptoms of getting sick. So because scientists were learning about how bats don't seem to express the genes, the inflammatory genes, when they're exposed to certain viruses, it began, causing them to ask the question, well, what regulates gene expression in all mammals, especially in humans? So this just kind of led to that, this discovery, that bat immune systems are different, from human immune systems. Excuse me. Okay, so what we have learned about eukaryotic regulation is that there are 5 primary levels of regulation or control. Let me get the highlighter so I can highlight some of this stuff. Oh, yay. Okay, sometimes it doesn't work right away. I have to play with it. So there are 5 levels of control, and the first 3 occur in the nucleus. So we call them nuclear levels. So these levels are chromatin structure. So this is going to be either the chromatin is in a form called heterochromatin, which cannot be transcribed. Excuse me. Or it is in a form called euchromatin, which can undergo transcription. So depending on which form the chromatin is in, euchromatin or heterochromatin, it will either undergo transcription or not. So that occurs in the nucleus, of course, because chromatin is found in the nucleus. Then there is transcriptional control, And transcription, remember, is when DNA codes for messenger RNA. So there's control of that process, and that is the most important of all of these control processes. Transcriptional control is the most important one of all these 5 different levels. The last 2 occur in the cytoplasm, and they're called translational control and post-translational control. Translation is when messenger RNA attaches to a ribosome and codes for the amino acids in a protein. So the 3 levels that occur in the nucleus are chromatin structure, transcription, and post-transcription control. And then in the cytoplasm, it's translation and post-translation control. And this is showing you the a cell's nucleus here and the pores of the nucleus where the messenger RNA leaves the cell. And then this is showing you a messenger RNA, polypeptide here, and this is showing a protein, which is basically the polypeptide that has undergone shape changes. But anyway, let's start with the chromatin. All right, so chromatin is DNA wrapped around histone proteins. They're the histone proteins there. So DNA wrapped around histone proteins. So if the chromatin is packed very, very tightly, then it cannot interact with RNA polymerase. And RNA polymerase is the enzyme that creates the messenger RNA. So if the chromatin is in the form called heterochromatin, it cannot undergo transcription. If transcription does occur, transcriptional control has to do with the splicing of certain introns, the removing of certain introns and splicing together of certain exons. We call that messenger RNA processing or alternative messenger RNA splicing. All of that goes under transcriptional control, and post-transcriptional control has to do with the addition of the cap and the tail to the final messenger RNA transcript, because that will determine the rate at which messenger RNA leaves the nucleus. It'll determine the life, the amount of time that the messenger RNA remains alive, or it's not really a living thing, but remains able to be translated into a protein. So its lifespan. And then translational control has to do with the production of the protein. Post-translational control is control that acts on the protein once it's already made. Okay, so chromatin structure, we said that DNA and histone proteins make up chromatin. DNA plus histone makes up chromatin, and the level of chromatin packing is going to either make it euchromatin, and if it's in this form, it can be transcribed into messenger RNA. So we say it's transcriptionally active. It's loosely coiled DNA, and it's transcriptionally active. But if it's in a form that's packed very tightly, it's called heterochromatin, and it is packed so tightly that it's transcriptionally inactive. So this is what we mean by the chromatin structure level of control. Now, an example of heterochromatin is something called a Barr body. Females of all mammals have 2 X sex chromosomes, but only one is active. One is in the form called heterochromatin, and it's inactive. So the inactive X chromosome is called a Barr body. And this is true for all female mammals. But a good example where we can see this and understand it is the calico cat. And a calico cat, if you didn't know this, is always a female. That calico coat pattern where the main fur is a cream color, but then there are patches of black and orange throughout. So what happens is the patches of orange and black in the calico cat's coat depend on which X chromosome in those fur patches is active. If you look here, one X chromosome has the orange color and the other X chromosome has the black color. So during cell division, if the black chromosome is active, if it's in a form called euchromatin, then that will be expressed as black patches of fur. And if the orange chromosome is active, it's in a form called euchromatin, then it will be expressed as orange fur. The inactive X chromosomes or bar bodies are in the form called heterochromatin, and you can see that they are shorter and they're packed more tightly together. So they will not be expressed. So histone modification, the histones, remember, are the proteins that the DNA is wrapped around. So DNA plus histones produce chromatin. And the histone modification determines if the chromatin is packed together very tightly as heterochromatin or if it's packed together loosely and as euchromatin. So this may be linked to something called epigenetic inheritance. Epigenetic inheritance, we talk about that. There's so many examples of it, but basically, okay, sorry, let me fix this so I can use the highlighter. Okay, but basically what happens is your genes code for certain traits, but if something else determines the inheritance of that gene besides just the DNA in that particular gene, then we call it epigenetic inheritance. Sometimes there's actually the presence of a gene that you have to have the presence of a different gene in order to express another gene. That's what we talk about sometimes with chapter 11, Mendelian genetics. I don't think I really gave a good example of that during chapter 11. But anyway, epigenetic inheritance is any time that variations in the pattern of inheritance, and that means, different alleles and different variations in how they're expressed, are not due just to the sequence of the DNA. So different variations in the traits are not only due to the alleles that you inherit. There are other reasons why you would have certain traits. So this is a better way to say it. I like this way. Epigenetic inheritance refers to inheritance patterns that are not dependent on the genes themselves. They have a really good example in this Smart Book of how exercise influences our cognitive function or how well we think. People who get regular exercise, in this case it was mice, they experimented on mice, but mice that get regular exercise express the genes for cognitive function differently, and they're able to actually, they're smarter, they're able to think better than the mice that don't get regular exercise. So that's an example of epigenetic. And then another example of epigenetic inheritance is genomic imprinting.